Physiology SAQ21 ADH. A. How is the antidiuretic hormone formed and released into the circulation, and how is this controlled? ADH formation. ADH or vasopressin is a nonapeptide with a molecular weight of 1000 daltons. It is synthesized by the magnocellular cells of the supraoptic and paraventricular nuclei of the hypothalamus. ADH release. The secretory granules containing ADH travel down the neuronal axons in the pituitary stalk to reach the posterior pituitary. The ADH neurophysin complex is released from the posterior pituitary into the bloodstream, and it dissociates immediately. Control of ADH secretion. ADH is secreted from the posterior pituitary into the systemic circulation in response to several stimuli, which includes increased plasma osmolality, reduced plasma volume, and other stimuli such as the stress response, nausea and vomiting, hypoxia, hypercapnia, acidosis, hypoglycemia, exercise, IPPV, chemical mediators, and drugs. Increased plasma osmolality is detected by osmoreceptors at the hypothalamus, located in the OVLT and SFO, which are two of the sensory circumventricular organs of the brain. They lack a blood-brain barrier, and this allows them to directly detect the tonicity of blood. Subsequently, water exits the cells and moves down its concentration gradient into the plasma. This stimulates the osmoreceptor cells to contract, which results in efferent signals being sent from the hypothalamus to the posterior pituitary gland to increase the release of ADH. ADH responds to a 2% change in osmolality with a threshold starting at 280 milliosmol per kg. Reduced plasma volume also increases plasma sodium concentrations, which increases osmolarity. Reduced plasma volume reduces MAP and reduces stretch of baroreceptors. Low-pressure baroreceptors and high-pressure baroreceptors are stretch-sensitive mechanoreceptors. The low-pressure type is located at the walls of atria and great veins. Reduced CVP and cardiac distension reduces the inhibitory efferents via cranial nerve 10. High-pressure baroreceptors are located at the carotid sinus and aortic arch. Hypotension reduces the inhibitory efferents via cranial nerve 9 and 10. This increases sympathetic tone, and this stimulates the release of ADH from the posterior pituitary. Conversely, a hypervolemic state reduces the release of ADH. ADH responds to a 10% change in blood volume, and the stimuli of change in blood volume overrides the stimuli of change in plasma osmolality. ADH secretion is inhibited in response to several stimuli, such as reduced plasma osmolality, increased plasma volume, chemical mediators such as ANP, opioids, and GABA, and drugs such as alcohol. B. What effects does ADH have in the kidney? Increased water reabsorption. V2 receptors are activated by ADH at the basolateral membrane of the principal cells of the cortical and medullary collecting ducts. This increases activity of adenylyl cyclase, which increases CAMP. This increases pKa, which causes insertion of aquaporin 2 channels to the apical membrane of the principal cells. This increases water reabsorption. This reduces the volume of urine and increases urine osmolarity. ADH also increases sodium reabsorption at the collecting duct by increasing the activity of epithelial sodium channels. ADH increases urea reabsorption via V2 receptor activation. This increases medullary interstitial osmolarity and promotes water reabsorption. ADH also reduces renal medullary blood flow. C. What biochemical abnormalities occur with excessive antidiuretic hormone secretion? This produces inappropriately concentrated urine, which may exceed 100 milliosmol per kg, euvolemic hypoosmolar hyponatremia. Serum osmolarity may be less than 280 milliosmol per kg, and serum sodium less than 135 millimoles per liter. Urine sodium concentration is usually increased and is more than 40 MEq per liter. Additional information. ADH, metabolism and excretion. ADH is rapidly metabolized by tissue peptidases, and about 1/3 is excreted by the kidneys. ADH has a biological half-life of 16 to 20 minutes. ADH receptor types. This includes V1, V2, and V3 receptors. V1 receptor is a GQ-PCR and is located at vascular smooth muscles, myometrium, and platelets. Actions include systemic vasoconstriction, platelet aggregation and degranulation, and pulmonary circulation vasodilation. V2 receptors are GS protein-coupled receptors and are located at distal tubules and collecting ducts of kidneys and endothelium. Actions include increased water reabsorption, increased sodium reabsorption, increased renal medullary blood flow, and increased factor VIII and von Willebrand factor release from endothelium. V3 receptors are GQ protein-coupled receptors. Actions include ACTH release as a neurotransmitter or mediator involved in memory consolidation or retrieval, and body temperature regulation. Vasopressin also has equal affinity for oxytocin receptors as oxytocin. What cell membranes in the body are not permeable to water? This includes bladder epithelium, ascending limb of loop of Henle, and cortical and medullary collecting ducts in the absence of ADH. What are the uses of ADH derivatives in critical care? Vasopressin is used as a vasopressor, for example in septic shock. Vasopressin is presented as a synthetic version of ADH. It is metabolized in the same way to endogenous vasopressin and has a half-life of 24 minutes. Cranial DI is treated with desmopressin, which is a synthetic analog of arginine vasopressin. It has 10 times the antidiuretic action of vasopressin, but 1,500 times less vasoconstrictor action. It has a half-life of 158 minutes. Bleeding esophageal varices are treated with terlipressin. This is triglycyl lysine vasopressin. Arginine is replaced with lysine at position 8 and has 3 glycine residues at the beginning of the peptide. The 3 glycine residues make terlipressin a prodrug. In the body, these are enzymatically cleaved by endothelial peptidases to produce lysine vasopressin. It has an elimination half-life of 50 minutes, effective half-life of 6 hours. Dose is 2 mg 4 hourly. Cochrane review in 2003 found terlipressin produced a relative risk reduction in mortality from varicose hemorrhage of 34% compared with placebo. Desmopressin is also used to treat coagulopathy and platelet dysfunction, such as in certain types of von Willebrand disease, mild forms of hemophilia, and impaired platelet function due to drugs such as aspirin or renal failure. This diagram from the Calgary Guide describes the pathophysiology of SIADH. Thank you.